Okay, so with DNA methylation over, our next topic is to talk about nucleosome positioning. So we go down this figure and see here, DNA methylation directly happen on the C nucleotide of the DNA, and then the DNA can form some structures. This is the double helix, and you can see there's a nucleosome. You can think about this as kind of a bead, and the DNA wraps around it about 2 circles, and then move on. This 2 circles is about 150 nucleotide long. And then we'll move on, and then there's another one, and it will move on, and so on. So in our genome, if you open the chromatin, also by the way, every time you talk about chromatin, it means it's DNA with all the proteins and all the packaging involved. The basic unit of the chromatin is this nucleosome. And the bead can kind of slide around and can be off and can be assembled again. And you can think about this like this. The inside, so remember there are 2 circles of DNA wrapped around, and inside there are 8 different histones. It's 2H2A, 2H2A, 2H2B, and then 1H3 and, 2H3 and the 2H4. So 4 different colors, and they are stacked on top of each other. And you can imagine this is kind of a Oreo cookie of 8 different proteins stacked on top of each other. And this region is called the nucleosome-free region, whereas this region is called the nucleosomal DNA. Okay, so this is nucleosome-free, this is nucleosomal DNA. And they are also very important because very often transcription factors, most transcription factors like to bind to the nucleosome-free DNA. If it's a nucleosome DNA, only a minority of transcription factors can really bind there. And if they do bind there, there are some transcription factors that are specifically bound there, and they will squeeze out the histones to make it open. So those are very often called the pioneering transcription factors. So they have to go there first to open up that region, squeeze out the nucleosome, then other transcription factors can go to bind. Because there are transcription factors that can only bind to the nucleosome-free region. And so if this region is bound by a nucleosome, then that's off limit to the transcription factors. Okay, so there are also experimental assays involved that can give us nucleosome occupancy, and that assay is called MNase sequencing. So MNase is microcalca nuclease. It's an enzyme that it's an endonuclease, which means it can cut in the middle of DNA, not just chew from the end. It cuts in the middle. Where does it like to cut? It usually likes to cut to the on the nucleosome-free regions. And once it cuts, it will also chew a little bit on the end until it reaches the histone. And so you can imagine if a MNase will cut in here, it will chew to here, it will also chew to here. And then if it cuts there, it will chew to this or chew to that. Okay, so basically, if you use MNase to digest your chromatin, what you're going to get is most of the nucleosome-free regions will be gone. You will be left with just the pearl and the DNA wrap around the pearl or the string. Okay, or wrap around the bead. And so after you use the microcalca nuclease to digest the chromatin, you usually want to run them in a gel. Once you run the gel, what you will see, you run the DNA you isolate the DNA, you run them on a DNA gel. What you will see is usually they come in letters, 1, 2, 3, 4, 5. It's because the enzyme digestion may not be complete if it is just a single nucleosome. Remember, this is about 140, 150 nucleotide long. The very first band you see is a DNA band that's about 150. But sometimes the second band is when you have a cut in here, you have a cut in there, but you didn't cut in the middle. So that piece of DNA encompasses 2 nucleosomes and the nucleosome-free DNA in the middle. And then the third one will be 3 nucleosomes cut on the outside, but there are 2 linkers. So these linker sequences are not cut. Right, so that's then you have 3, you have 4, and very often people just cut out the mononucleosome to do sequencing. And you can do paired end or single end, either way, you can sequence them out. This, again, it was a study, review paper in 2009. And at that time, sequencing is super expensive. People are still only sequencing just one end of the fragment. Nowadays, we just sequence both ends. It's fairly easy. But so you can imagine if the nucleosome is binding on the DNA and you are using an enzyme to cut and chew, you will chew all the way kind of close to the end, and then the DNA will wrap around 2 circles for about 150 nucleotides, and you can link the adapter to sequence it out either from the left or from the right. So this kind of is similar to a ChIP-seq experiment, because this is just a protein binding to the DNA, right? And so you will see a bunch of reads that's mapped to the positive strand, and you'll see a bunch of reads that's mapped to the minus strand, and usually in the middle of that is a nucleosome. And so you can do that, and you merge them, kind of you shift them each to about 75 nucleotide, because the nucleosome is about 150 long. And you know you are able to sequence it either from the left or from the right. And so if you shift 75 to the middle, this is where usually that nucleosome is. Okay?
So if you see multiple landing patterns, why don't you just continue to digest?
Your question is, yeah, why don't you just digest that? By the time you run the gel, it's already in DNA. The nucleosome is gone.
Oh, so you
Yeah, after you finish the digestion, you isolate the DNA to run the gel. And you just, like at that time, if you digest the second band, you will chew up everything, right? Because the nucleosome is gone already.
I assume that's part of
It's okay. You get a good enough representation. People don't really worry too much about those. There's always something that failed to digest. Okay, so yeah, this is a very easy experiment, actually. The cost is mostly sequencing. The digestion part is very easy to do. Okay, so with that, people are trying to look at what are some of the properties for transcription factor binding. And so they want to see whether we can computationally predict where the transcription factors can bind. And in this 2006 paper, they try to use computational method. And what later on in the field we discovered is that there are 2 levels of nucleosome positioning. One is called a rotational positioning. So if you look at the mammalian genome, so by the way, bacteria does not have nucleosome. Yeast is, when you start to have eukaryotes, is when you have the genome. And if you look at the yeast or chicken or human, they notice that even though the trend is not super strong, but overall you can see this. The DNA actually have a 10 base pair periodicity in terms of its AT compared to a GC. And so usually, if you look at a piece of DNA on average, this is actually a very weak trend, but you do see it overall is there, for a little bit of the DNA, there's a bit more AT, and then about 10 base pair away, it will be a little bit more GC, and then a 10 base pair away is a little bit more GC and AT and GC and AT and so on. So the reason for that is you can think about this. Sorry. So remember, the DNA is a double helix. It turns, and as it turns, it also have to wrap around the bead. And so as a result, so these are the histones, and this is the DNA. And remember, yeah, so the DNA actually turns, and the one turn of the double helix is about 10 nucleotide. And so in another 10 nucleotide, it will be kind of an opposite side, so on the strand that is facing the histone. And so it turns out the GC likes to interact more with the histone in the middle. The AT does not. In general, the histone in the nucleosome, basically those beads, they do not like GC. They like, sorry, they like GC, they don't like AT. And so over evolution, DNA in the species are evolved so that you have this kind of periodicity in every 10 base pair. This is to make sure that it's always the GC that faces the histone inside, or at least it favors that. And so when the nucleosome is not in place, it's like everybody is sitting on something not so happy. But if you just slide a little bit, then it's really snug, everything fits, right? So, and so using this, you can help decide whether the nucleosome should be here or the next base or the next base. And so with this, you can actually predict where the nucleosome will form stably. When you are a little bit off, then everybody is unhappy. So even though the trend for this AATT 10 base pair periodicity is very, very weak, but because you have 150 base pair that you try to fit together, eventually you will make the nucleosome fit more snugly in some locations than other locations. So this is called a rotational position because you're only kind of trying to find a snug fit in this location. There's another nucleosome positioning which is called statistical positioning or translational positioning. It really is asking, is the nucleosome here or there, or really far away, the center of that nucleosome, where that is. And if you sequence a lot of cells, you will see that, for example, in locations when nothing is happening, the nucleosome in the different cells, they really don't care. So because you're sequencing the nucleosome from many different cells, you can see they're out of sync, so you don't really get a very good signal. But if some locations, for example, the transcription start site, you need a polymerase really to recognize the right place, it binds there, and once it's there, the next nucleosome that can come in is pretty much all well aligned in all of the cells. Therefore, in this location, you see a very strong translational positioning because this big gap is where the transcription complex is sitting. It's blocking the things. And the +1 and -1 nucleosome is very well positioned. And because they are positioned, their next one also cannot shift as much left and right. They are also fairly positioned. The next one is positioned. It's like when you have the Olympic run, initially say go, they kind of are all pretty close. But then over time, you gradually lose the things in sync. But the very beginning of the gene, all the cells in that location have the +1 nucleosome, +2 nucleosome, +3 nucleosome, and then they gradually goes weak. And so this is actually scientifically more interesting because when you have transcription factor binding in the enhancers, very often that region also have a statistical or translational positioning or nucleosome-free region where the transcription factor is occupying the site and it's squeezing out the nucleosome. And right next to it, you will see +1, +2, +3. They are fairly well positioned, but eventually you also lose the positioning gradually. Okay, and so basically DNA methylation and nucleosome positioning are just different aspects of epigenetics that create landmarks. So both nucleosome and the DNA usually prevents transcription factor binding. And therefore, in order for transcription factor to bind, you do not want DNA methylation, you do not want the nucleosome. You want it to be free and open. Okay, that's all for today.